In this paper of bioorganic and biophysical chemistry, you will be learning various applications of chemistry in biology. These would include the structure and reactions of various biomolecules such as DNA, proteins, vitamins, as well as coenzymes. You will also learn about their locations within cells. You will also be learning how biomolecules are purified and characterized using biophysical and biochemical techniques. A detailed analysis of enzymes and their mechanisms will also be described. Furthermore, the application of the study of enzymes in therapeutics, diagnostics and in drug design are also being dealt with for your knowledge. In this module, you will be learning 1. Living organisms consists of cells. Different types of cells have different functions, so you will be learning about them, and you will also learn how cells are visualized. Introduction. The basic unit of a living organism is a cell, whether it is a bacterium, a plant or an animal. Unicellular organisms such as bacteria, yeast and some protozoans like amoeba have only one cell which carries out all the living functions of the organism such as feeding, growth, use and generation of energy and reproduction. Plants and animals are multicellular organisms which have groups of cells specialized for certain functions but are coordinated in their activities to benefit the successful propagation of the organism in a given environment. Cells consist of a myriad of molecules present in a milieu of water. These molecules interact with each other to perform the basic functions of life such as growth and reproduction. If these biomolecules were taken into a test tube, they would hardly show signs of life, and hence the organization of the cell is essential to regulate the interaction of these molecules so that life is perpetuated. This module will introduce you to the architecture of various cells which are unique to the organisms they are part of, and also introduce you to techniques used to study these features. Cell structure and techniques to study them in prokaryotes and eukaryotes. The basic unit of an organism as diverse as a bacterium or fish or man is a cell. A cell consists of various biomolecules in an aqueous solution called cytoplasm, enclosed or bound by a lipid layer and frequently in addition by a tough wall called cell wall, which protects the cell from the environment. Based on the features within the cell, there are two types, prokaryotes and eukaryotes. Prokaryotes are simpler in structure, having their genetic material enclosed only by the cell envelope. Bacteria fall into this group and are usually called single-celled organisms. The second group, called eukaryotes, have additional compartments within their cells enveloped by a single or double lipid membrane. These compartments are called organelles. The genetic material in eukaryotes lies within the confines of the nucleus, which has a double lipid membrane. This is the basic difference between prokaryotes and eukaryotes, wherein in the latter the genetic material is enveloped. It is thought that during early evolution, prokaryotes engulfed other prokaryotes, leading to larger eukaryotes with membrane-bound genetic material which formed nuclei and other organelles, notably mitochondria and chloroplasts which have a double membraned enveloped structure. This theory is known as the endosymbiont theory. The average diameter of a prokaryote is about 1 micrometer, whereas that of a eukaryote is about 100 micrometers. Let us look at the structures of both prokaryotic and a eukaryotic cell. Some of the features of the prokaryotic cell are as follows. There is a cell wall, there is a plasma membrane enclosing a cytoplasm which has various other subcellular kind of particles like ribosomes, plasmids, as well as sometimes also you have certain vacuoles. On the other hand, when you look at a more advanced eukaryotic cell, you find that many of these smaller subcellular particles which were present in the prokaryote are actually being enveloped, and therefore you have what are known as organelles. So, in addition to a cell wall, plasma membrane, a eukaryotic cell also has a number of organelles. For example, if it is the chromosomal DNA, it is present within an envelope which is known as the nucleus. Similarly, when you have certain proteins and certain lipids, they are enclosed in certain vesicles such as the endoplasmic reticulum. And therefore, the big difference between a prokaryote and a eukaryotic cell is that a eukaryotic cell has many more compartments known as organelles. Now, let us look at the eukaryotes and their organelles. Eukaryotes consist of single cell eukaryotes such as yeast and amoeba, as well as multicellular organisms such as plants and animals. The cells of eukaryotes contain organelles which include mitochondria, lysosomes, Golgi, chloroplasts, peroxisomes, endoplasmic reticulum. The functions of these organelles in the life of a cell are described as in the nucleus. This has the genetic material which is replicated to propagate the cell and transcribe into RNA to express genes and eventually proteins. The mitochondria are the powerhouses which generate ATP, namely energy currency, and break down sugars and lipids for the same. The endoplasmic reticulum is responsible for the synthesis of lipids and proteins for export. The lysosomes are organelles which are responsible for the breakdown of various molecules by hydrolytic enzymes. The Golgi are responsible for the glycosylation of proteins and also for sorting out proteins which have to be moved outside the cell. The peroxisomes perform oxidative reactions using oxygen and hydrogen peroxide. Chloroplasts, which are unique in green plants, generate sugar and carbohydrates, the process being photosynthesis, by splitting water using energy of sunlight. Vacuoles, which are present in plant cells, fungal cells and some animal cells, are used for storing water, salts and also remove molecules which may be toxic to the cell. Microscopy techniques are some of the most powerful techniques to visualize cells. The advent and the understanding of biology was only possible because of the invention of the microscope. The microscope, which uses basically light or ambient light, are known as light microscopes. Cells, whether prokaryotic or eukaryotic, are too small and cannot be seen by the unaided eye. They can only be viewed with the help of special instruments known as the microscopes. These generate magnified images of the cells. However, microscopes are also limited in their resolution. Resolution is the smallest distance between two points which can be seen by the wavelength of the radiation used to illuminate the sample. For example, The light microscopes which use visible light of wavelength 350 nanometers to 700 nanometers have a theoretical resolution limit of about 200 nanometers. In general, the theoretical resolution limit of a microscope is determined by the equation resolution limit is equal to 0.61 into lambda divided by n sin theta. Lambda is the wavelength of visible light, n the refractive index of the medium, air or oil separating the specimen from the objective and the condenser lens, theta half the angular width of the cone of light collected by the objective lens, maximum being 180 degrees and therefore sin 90 being 1. In other words, the resolution limit is approximately half the wavelength of the ambient light used in the microscope. Another example is the electron microscope, wherein an accelerated beam of electrons is used whose wavelength is 0.004 nanometers and therefore its resolution theoretically is about 100,000 times that of the light microscope. However, practically the resolution is only about 500 times that of the light microscope due to the aberration of the optical lenses used. Most prokaryotic and eukaryotic cells are visible by light microscopy. Observe the optical path in a light microscope. From a condenser, there is light passing through the sample and then through a set of lenses it reaches the eye. These images are therefore magnified depending on both the focal length of the objective lens as well as the eyepiece. Variants of the bright field light microscope such as phase contrast, dark field are selectively used for better picture quality of the sample. Why are these known as bright field? Because light passes through the sample and then is observed through a system of lenses by the human eye. In general, the principle of light microscopy involves illumination of a thin sample of cells before or after staining by light from a low voltage lamp suitably focused onto the sample by a condenser and collection of light passed by a system of lenses, objective and eyepiece or camera. The first one is a detailed picture of cells from a eukaryotic source, namely an animal source. You can clearly see a number of details in the cell and these have been enhanced by coloring, usually the cell with various dyes. Observe that the coloring improves contrast and therefore visibility of various parts of the cell. This picture, which has a greenish background, is that of certain bacilli. Bacilli are actually prokaryotic cells. They are much smaller, at least 100 times smaller than a eukaryotic cell which you have previously seen. Therefore, not much detail is available within them as far as light microscopic can discern. The darkish photograph which you see are basically neuronal cells observed with some lighting, and this is basically an example of dark field microscope. In dark field, illumination is slightly different from how it is in a bright field. In a bright field, the light passes through the sample. In a dark field, it is indirectly lighted up, and therefore the neuronal cells are sort of highlighted like a spotlight rather than the light passing through them. The cells picturized in this greenish kind of cells, which are 3 cells together, these are cells observed in a fluorescent microscope. In a fluorescent microscope, the ambient light is also visible light, but of course of a shorter wavelength, namely UV light, and because the proteins which have been tagged with certain fluorescent materials are lighted in the presence of UV light, they fluoresce. So, typically you see the green or yellow fluorescence in these cells. Fluorescence microscopy does not give you too much detail as such, but the presence of certain molecules, particularly like cytoplasmic material, cell wall proteins, etc., can be highlighted by this kind of microscopy. Light microscopy has some limitations, and that is basically resolution. Want to look at details within eukaryotic cells, or for that matter details even within a prokaryotic cell, then the resolution required is much higher, and this can only be met by the electron microscope. As already indicated to you, the wavelength of radiation used in a microscope has a great bearing on the resolution of a cell. Hence, in the electron microscope, as electrons are accelerated at voltages nearing 100,000 volts, the wavelengths can be as low as 0.004 nanometers. Small cellular structures such as details of organelles smaller than 0.1 nanometers and subcellular particles such as ribosomes can be visualized. The principle of electron microscope is similar to that of the light microscope, except glass lenses are replaced by electromagnetic coils. Another major difference is in the preparation of the sample. The cells to be visualized have to be stained by electron dense salts like osmium tetroxide and then have to be embedded into resins which solidify. Thin slices, typically 50 to 100 nanometer thick slices of the sample tissue are cut using a microtome and then placed on grids for viewing. In the transmission electron microscope TEM, electron beams are allowed to pass through these thin slices. Some of the electrons hitting the dense osmium atoms are scattered, others are gathered and focused by magnetic coils onto fluorescent screens which are photographed. In another application of the electron microscope, the surface characteristics of cells are visualized by a method called scanning electron microscopy or SEM. In this case, the sample surface is visualized by a method called scanning electron microscopy. The sample surface is shadowed by an electron opaque and stable shadow of carbon or platinum. The cell is then dissolved away by chromic acid and the shadow is placed on a grid and visualized. In the SEM, the electron beam, instead of going through the sample, is reflected by the shadow and the gathered electron waves are visualized on fluorescent screens. The path of the various radiations, whether it is light or electron beams, are well illustrated. In the case of light microscopy and transmission electron microscopy, the pattern appears to be similar except that the glass lenses are replaced by electromagnetic kind of coils which serve as the lens to cause the electron beams to condense. The sample or the ultimate viewing in the case of the light microscopy is the eye or a camera. Obviously, in the case of the electron beam, it has to be a fluorescent screen which is then of course photographed. In the scanning electron microscope, first of all, using various magnetic coils which form a part of the two or three condensers indicated, they fall on the sample surface, basically a copy of the sample surface in the form of a carbon or a platinum shadow, and then these are reflected and gathered by a separate fluorescent screen. LM stands for light microscopy in the picture, TM for transmission electron microscopy, and SEM for scanning electron microscopy. The light source In the case of the electron microscopy is a electron gun. Observe the picture viewed by TEM. Very easily you can discern the various parts of a organelle. An organelle, remember, is the size typically of a bacteria, and a bacteria, you know, or a prokaryote cannot be observed in the regular light microscopy, and therefore the electron microscope presents a higher resolution typically needed to view cells, particularly internal details of both eukaryotic cells as well as prokaryotic cells. Observe these other electron microscope or pictures. The first one is a TM image of a Golgi complex. As indicated to you earlier, the Golgi complex is an important organelle of a eukaryotic cell, which is important in both glycosylation of various proteins as well as targeting them or distributing them for presence within a cell or export out of the cell. So, they are involved both in transport as well as glycosylation. Details of these various parts of a Golgi complex are typically illustrated by this TEM or transmission electron microscope. The second picture is that of the mitochondria. The mitochondria are the powerhouses of eukaryotic cells. They generate the all important ATP required for all functions of a cell. In fact, ATP is also known as the currency or the energy currency of a cell. The mitochondria are very clearly discernible in this TEM image as a double-walled kind of organelle, wherein the outer membrane is smooth, whereas the inner membrane folds into cristae and therefore also increases the surface area of various kinds of important proteins involved in generating ATP from the reduction of oxygen. The other two pictures, first that of a blood cell is observed, and this is a typical scanning electron image, and therefore you can easily see how a scanning electron image is different from that of a transmission electron image. Here the RBC is seen as a biconcave structure. You see the overall surface of the cell. Beside this is another cell, perhaps that which is known as a white blood cell. Also present in the next picture, which is typically a SEM picture, you see the overall surface of a Drosophila. A Drosophila is also known as a fruit fly. Its eye is a compound eye, and you can easily see the surface contours of the eye. The next image is that of the pollen grains. The structure, the overall surface structure of the pollen grains can be easily seen in this scanning electron image. Note, the difference between a transmission electron micrograph or image and a scanning electron micrograph or image is basically the previous, that is the transmission one gives internal details, the scanning one gives the surface details. Another powerful method of microscope is the fluorescence microscope, which has become invaluable to most cell biology laboratories. In this type of microscopy, fluorescent tags are added to specifically label cells, organelles, proteins, and then these are viewed under the absorbing radiation. The fluorescence emitted is viewed through a filter against a dark background. Only the fluorescence emitting regions are clearly visible. The picture which you are observing is that of mouse cells, which are derived from the small intestines using fluorescent tags for proteins present. Very easily you can see the contours of the cells, the various parts of the cells, and therefore you can also sometimes reflect on the function of these. The confocal microscope is a specialized development of the fluorescent microscope, where the practical resolution achieved is around 200 nanometers. This technique allows one to image live cells and cellular processes if appropriately tagged with fluors. FRAP, which is an abbreviation for fluorescence reactivation after photobleaching, and FRET, fluorescence resonance energy transfer, are specialized applications explained in shortly. Using biochemical and molecular biology tools, one can fluorescently label specific proteins such as DNA, lipids, etc. One can also label several different species with different colored tags. The sample is then illuminated with the excitation wavelength of the fluorophore, and the emitted light is gathered by the objective as is done in any microscope to be viewed by the eye or camera. In the LSCM, which is laser scanning confocal microscope, the specialized feature is that the illuminated cell is scanned by a laser excitation wavelength point by point and then plane by plane with concomitant gathering of the emitted light simultaneously from only the focal plane at a given step confocal plane. The plane by plane images are stored in a computer and the 3D image is constructed. Observe the confocal image of mouse cells which have been stained with fluors targeted to nuclear and cytoplasmic proteins. The schematic diagram of a confocal microscope is indicated here. Wherever the line or plane of focus for the light beam is done, it is gathered from that same focus, and that is why you get a plane by plane image which is then stored in a computer and then reassembled in silico, and sometimes you can also color these with various colors so that the contrast is enhanced. Now let us see what FRAP and FRET techniques are about. When excited by high intensity radiation, the fluorophores in a region in the sample can be damaged, a process known as photobleaching, leading to a temporary loss in the fluorescence emission signal. However, in time, the fluorophores from the neighboring region will diffuse and recover the decreased fluorescence in the targeted region. This is the principle of FRAP. By this procedure, which uses a fluorescence microscope which can record fluorescence as a function of time, the movement of molecules in plasma membrane can be determined. In the FRAP procedure, which uses a fluorescence microscope with a recording facility, fluorescence is recorded as a function of time. The movement of molecules in a plasma membrane can be determined. The rate of recovery of fluorescence can then be used to study how the molecule being studied diffuses in space. One can then make estimates of the size of the molecule. Another technique, namely FRET, is also easily understood. The emitted energy of one fluorophore may be transferred to another light sensitive molecule through a non-radiative process. In other words, no photons are exchanged. The fluorescence emission of the first fluorophore will be apparently smaller or even quenched. This is the principle of the FRET technique. However, the quenching will be effective only when the two molecules are near each other in space. Use of the fluorophore quencher pair can be applied to various studies, say studying whether protein A interacts with protein B in some region in the cell. Now let us summarize what we have learnt in this module. All living organisms are made up of cells. Prokaryotes like bacteria are single-celled, with diameters around 2 micrometers, and eukaryotes like yeast, plants and animals have larger cells with diameters around 100 micrometers. Prokaryotes have a cell envelope only, but eukaryotes contain, in addition to a cell envelope, several membrane-bound organelles which conduct specialized activities such as energy generation, DNA replication, etc. The structure of the cell is a key to its functioning, and hence microscopy is an invaluable aid in studying cells. To enable the observation of different sized cells, a variety of microscopes have been invented. The resolution limit of any microscope is approximately half the wavelength of the radiation used. The light microscope, which uses visible light, has a resolution limit of about 200 nanometers, and hence structures smaller than this size are observed by the electron microscope, which has a resolution limit of about 0.01 nanometers. In both types of microscopy, samples have to be prepared and stained for better viewing and contrast. In fluorescence based microscopy, especially confocal microscopy, live cells can be viewed. In phase contrast microscopy also, live cells can be viewed and photographed using time-lapse photography, and events such as cell division, surface movement of molecules can be recorded.